Okay, hello everyone. So welcome to the second week of our special lecture series. My name is Ming Zhang. I'm a professor of statistics and also the associate director of data science at Purdue University Center for Cancer Research. So this seminar series is part of our NCI-funded summer workshop on big data training for cancer research. Today we have three panelists, John Fry, Nadia Lama, and myself. So John has helped with all the Zoom meetings and the registrations. He will also record the lecture and post the recording online after the seminar. So now I would like to introduce our webinar moderator, Dr. Nadia Lama. So Dr. Lama is a research assistant professor in the Department of Comparative Pathobiology at Purdue. She has been the manager of the Purdue side of the collaborative core for cancer bioinformatics since 2015. So she has a background in biochemistry, molecular biology, computational biology, and she actually got her PhD in 2015 from Purdue. So Dr. Lama's research interests include single-cell data analysis and data integration with a focus on immunological diseases of the prostate. So with that, I will hand it over to Nadia.
Thank you, Min. And welcome back, everyone. So just as we've done previously, you'll be muted for the duration of this seminar, and you can feel free to use the Q&A function on the bottom right side of your Zoom screen to ask questions throughout the seminar. And feel free to upvote questions that you like, and those will bubble to the top and have a greater chance of being answered. So now with that, I'll hand it back to Min.
Thanks, Nadia. It's my great pleasure today to introduce our speaker, Dr. Mark Kelley. So Dr. Kelley is Associate Director of Basic Science Research at Indiana University Simon Comprehensive Cancer Center, and he's also the Betty and Earl Her Professor in Pediatric Oncology Research and a professor in the departments of Pediatrics, Biochemistry, Molecular Biology, Pharmacology and Toxicology, and Ophthalmology. He's also a joint professor in Eugene and Marilyn Glick Eye Institute and the director of program in Pediatric Molecular Oncology and Experimental Therapeutics. Among many of his titles, I really want to take this opportunity to thank Dr. Kelley for his great support for our workshops from the beginning when we started to write the proposal 3 years ago. I think we probably most of you have met Dr. Kelley in the opening remarks, and today, so he will speak on some of his research, which really focuses on translational research in DNA damage and repair, specific to determine how these activities can be exploited therapeutically to treat cancers and protect normal cells from oxidative and DNA-based damage. And among all the discoveries during his career, so have culminated in 21 patents and over 190 articles in peer-reviewed journals, and as well as 31 book chapters. So I think since 1993, he has a focus on specific on enzyme, purinic, I think it's called the redox effector 1. I just called it Ref1. It seems to be a long name. Remember last year, Dr. Kelley gave a really great story on Ref1 already. And I think at least for me, I'm excited to hear, how the trial going on and more update about the Ref1. So with that, so please join me in welcoming Dr. Kelley.
Thank you very much. Thanks for that nice introduction. And I enjoyed this workshop and working with our Purdue colleagues on this, particularly Min, Nadia, John, and others who've put this together and made it very easy for us to participate in this. I'm going to share my screen. And does this look okay? Good. So, sorry, it's blocking some of it. So I'm going to today kind of take you on a long and winding road, a trip with a little science and talk about this target, APE1, Ref1, which is shown right here in the title and I'll talk about more in a minute. So a little bit of science, but I want to talk about various things we've done to translate our findings from the bench to the clinic. And these are just my disclosures. I do have, I founded Apexian Pharmaceuticals, and we have a license agreement with Occupier, and that'll explain in a little bit why I'm an adjunct in the Glick Center for Eye Research in the Department of Ophthalmology. Are these slides advancing okay? I just want to make sure. Okay. So like I said, I'm going to talk a little bit about science, and I'm just going to give you some snippets of the science. I'm going to talk to you about how we're getting more and more into the big data bioinformatics. Actually, Nadia was in on our first paper studying this protein and some of the data we had, how we went from the pathway from the bench to a trial and future trials, and where we're going with all this, and really a lot of really cool new areas we've discovered because of our collaborations with the various bioinformatics, both here and at Purdue. So what I'm going to talk to you about today is this protein called APE1-Ref1. APE1 stands for apirintic endonuclease 1, and it also has the name of redox effector factor 1. And there's only a Ref1, there's no Ref2. There is an APE2, but it doesn't have the same function in humans. So they're really a 1 and a 1. And actually, this is a funny story about this. I can talk about if we have time. So for years, and I figured now that I've been studying this protein for over 30 years, probably a little longer. And I started out as a DNA repair guy studying this DNA, the repair of DNA damage. But it was discovered then later on that the same protein, APE1, has this other function of this redox effector factor signaling, which is basically converting oxidized transcription factors from an oxidized state to a reduced state and taking them from an off to an on status. The other thing I'm going to talk a lot about today, and I'm going to just introduce it now, is a drug called APX3330 shown here. This is a drug that we took to phase 1 and now phase 2 clinical trials. And I'll talk more about that in a minute. So APE1-Ref1, I mentioned, has two major functions. One is this DNA repair, and the other is the redox. The interesting thing about this is you can mutate either function and not affect the other. So we have made protein that's redox active and repair deficient and redox active and vice versa both ways. And so we actually are still heavily involved in figuring out the exact mechanisms of some of this, but it makes it a very intriguing molecule because of these dual functions. The repair function, as I introduced a little bit, happens if you have a damaged base. So this is APE is a major player in the pathway of DNA base excision repair. Do I have a pointer here? Is this a, yes, laser. There we go. Oops, didn't work. Oh well, in base excision repair. And what happens is if you have a damaged base, it gets removed, and APE comes along and makes a cut, and then it gets filled in. And this is the only enzyme that does this, so it's very critical in the cell to make this happen. It also works on another part of the pathway. If you have an oxidized base that's damaged and the base gets removed, APE makes the cut, polishes the end so it can complete and ligate the DNA back together. I've spent a lot of my life working on this, and we're still doing these studies, but we've been focusing more on the redox function lately. So it's an intriguing molecule with more of the redox active function here at the amino end, and then the repair domain more at the carboxy end. And like I said, you can make mutations in some of these cysteines up at this end, and that'll kill the redox activity, or you can make mutations in some of these other amino acids and kill the DNA repair activity. And it is also found in the mitochondrial for DNA repair, which we'll talk about. So I'm going to spend most of the day talking about this redox function activity. So we were working on APE-Ref1, like I said, back in the late 80s, early 90s. And in the mid 90s, the end of the 90s, there was a protein discovered that converted transcription factors from an inactive state, okay, to an active state. And this was discovered by Tom Curran, who was at that time at St. Jude's, who then moved up to the East Coast and now actually is over in Kansas. And that's why Tom thought when he was studying this that he would find multiple factors that would do this redox activity. So the first one he named REF1. And I actually had a call with Tom just the other day, and there's no REF2, 3, 4, or 5. And so we joke about that it's REF1, but it's the only REF. Well, it turns out back in those days that you actually purified protein in the cold room, and you did your biochemical activity assays, and then you cut up your protein and you got it sequenced the amino acids, and then you went and looked for the DNA. So it's very, very old school. And when they finished all that, the exact sequence was APE1, a protein we knew that is involved in the DNA repair. And then so what followed were a few years of people arguing whether this was real or not, was it a different protein and all that, and it turns out it is. And so this protein has two major functions and actually some other minor functions, but the major ones are this DNA repair and this redox signaling. And so what we've been focusing on is blocking this redox signaling a lot. So our compound I'm going to talk about today is 3330, and I'm also going to talk to you about two new compounds we made, Apex 2009 and 14, and we have another half dozen or dozen more that we're studying. And what it does is it blocks through protein-protein interaction, the protein-protein interaction. It basically converts it blocks APE from converting it. Usually it's a 2-electron transfer to the cysteines from APE to the transcription factors, and it blocks that so they can't be turned on. When the transcription factors are on, and the targets originally found and still found that are important factors like NF-kappa B for inflammation, FOS and Jun, AP1, CREB, STAT3 for inflammation, and importantly, HIF-1α, which turns on VEGF for angiogenesis. Well, all of these are important in cancer, and as I'll show you, they're important in some other diseases too. So if you add 3330 or the other compounds, it blocks this, and then so what happens is these activities are turned off, and so these transcription factors can no longer bind to the DNA and can't turn on these downstream genes. And so in cancer cells, where we've looked at it a lot, and I'll show you a little data, the redox function is really necessary because the cancer cells are revved up, they're dividing, and a lot's going on, and they're responding to various stimuli. And we know that under hypoxic, most solid tumors are under hypoxic conditions, HIF-1α levels are up. You have STAT3 for the JAK-STAT signaling pathways, NF-kappa B and others. And so what happens is the tumor cells really needs this activity to turn on these transcription factors. How does this happen? Well, we have some models, and the publications are on the bottom, but it turns out that 3330 interacts with a cysteine 65, which is buried in the protein, and interacts with it and converts it from a reduced to an oxidized state, which makes it inactive. We also know the protein unfolds for this buried cysteine to be exposed to interact with the downstream transcription factor. There's two other cysteines involved, 93 and 99, that I'm not going to show here, that are involved in these disulfide bonds between the proteins. So what's fascinating, though, and we actually have some recent data we're just getting, is a little bit more on how this mechanism is interacting. And I think we're going to have some good stories come out. Maybe if I get asked to talk next year, we'll have some good data on some of this redox signaling, but it's a fascinating process that's occurring. Well, we also made some new generation of the compounds through my company, Apexian Pharmaceuticals. We had a number of really good chemists, actually ex-Lilly chemists, lead chemists and scientific board directors. Homer Pierce, he led Lilly Oncology for many, many years. And by having this group of chemists, we were able to take the parent compound and do a SAR around it and develop, which actually a 4, 5, 6 step synthesis a bunch of new molecules. And the way we assay these is you take a purified APE that's reduced, and you take a target. This could be NF-kappa B or FOS or Jun or HIF-1α with the DNA binding, and you show that when you add 3330, you block the ability of APE to reduce the transcription factor, and with increasing concentrations, you get less and less binding on this. We call these redox heme cells, the gel shifts, and you see that 2009 is much more potent, and 2014 are more potent. So what I'm going to talk about today is kind of APE-Ref1 is the center of the universe with these multiple functions, both in redox and repair. This is actually a figure that's in a review article that's coming out soon. And we're going to talk about how blocking this redox function has implications for inflammatory bowel disease, inflammations for anti-CIPN, chemotherapy-induced peripheral neuropathy, some new data we've just about to publish on in cancer metabolism. And what's not shown here is some data on the eye that I'll talk about a little bit later in our my talk. So you look at this and you say, well, geez, Mark, you're really unfocused. You're all over the place. How can you be doing this? How can your drugs be doing all this? How can your protein be doing all this? Well, we never lose sight that we're focusing on APE-Ref1, its basic functions, and then blocking the particular function. Now, what we are focused on then is, okay, so what other disease indications fall in our wheelhouse that would have inflammation and angiogenesis? And that is the eye, turns out inflammatory bowel disease, and then of course cancer. So even though we may look unfocused, we're still focusing on this major activity. We're just looking at the downstream from that. So I'd like to show you just a little bit of data on pancreatic cancer and bladder cancer, and a little bit on this AML data we had. And then we'll talk about some of these other things later through the talk. And I apologize for the brevity of the data, but I'm more than happy to discuss it or send references if you'd like. So for years, we've been studying pancreatic cancer, and we've been looking at, APE1-Ref1. Well, we did a lot of single agent studies using the various inhibitors. And because STAT3 is one of the targets of Ref1, actually STAT3 was found to be under redox control. And then my collaborator, Dr. Melissa Fischel, she's the one that actually published and showed that this redox control is by APE1-Ref1. And a lot of people have been trying to target STAT3 directly. And it's part of the JAK-STAT signaling pathway. Well, there's drugs out there like ruxolitinib that block JAK in the JAK-STAT pathway. And there's a drug that's actually been in phase 1 and phase 2 trials called napabucosin that's shown to block STAT3 activity, but not its redox or phosphorylation activity. So some of the things we did, I'm just going to show you a little data, but we've been looking at combination data, because I think all of us know that most tumors, particularly solid tumors, you can't hit them with one agent. You need to block multiple pathways. And so we got studying this, and we thought, well, what if we used our inhibitors to block the redox function of REF to keep STAT3 from binding, and combined it with napabucosin that attacks a different point in this pathway. We did some work with our inhibitors with ruxolitinib and saw very nice synergy, actually, by blocking up here with JAK and then STAT, but I'm not going to show you that today. So some of the studies we do is, and Melissa's an expert on these 3D tumor modelings, where we take either the tumor alone, and these are patient-derived tumors, or tumors plus cancer-associated fibroblasts in 3D culture. And so we do these, and the tumors are labeled red, and the calves are labeled green. One of the things in pancreatic cancer has been a big discussion, and there were a number of papers a few years ago showing if you kill the cancer-associated fibroblasts, you actually make the tumor worse. And so you really want to look for agents that are targeting the tumor more than the calves. And that's one of our rationale for screening our molecules, looking for tumor killing versus the tumor in the calf killing. And as you can see, when we do this over a number of days, there's a 3D cultures, and you can see 3330, you have to use fairly good micromolar amounts. I will tell you that you can get up to 150 micromolar levels in human serum. So it is doable, and I can discuss why you need so much. But you can see we get killing at this level in the tumor alone or tumor in calves, and they're a little more resistant with the calves. You can look at 2009 and you can use less to get the killing and actually get better killing when you have tumor and calves together. And then here with 14, even at a much lower level, you're getting really nice killing and a nice dose response through this. That's as a single agent. And this is really a paper that just came out, came out in 2020, December. And this is where we then started doing single agents, but then we started adding naprobucosin with this for combo. And so in all these 3D spheroids, again, they're patient-derived spheroids with the tumor in red and the calves in green. What you can see is control, single agent, some killing, and it depends on, of course, the concentration, 9, some more, and 14, you get a lot of killing. When we do the combo with NAPA plus 3339 or 14, NAPA gives you some killing kind of here. 33 combo, you get much better. 9 or 14, you're really wiping out the tumors and not really touching the calves. So we're very excited about this data going forward, because this is and this is all quantified for calf intensity versus tumor. So we're excited that these sorts of combos may be targeting the pancreatic tumor cells and leaving the microenvironment, the calves alone. So obviously more work has to be done on this. One of the other things we did is looking at how we're affecting this pathway, the JAK-STAT pathway. And so we looked at whether naprobucosin or ruxolitinib affected the phosphorylation of STAT3. And as you can see, it doesn't really do anything. You don't get a change in STAT3 levels, and you really, and you do have an effect. You can see here with the naprobucosin and rux, you're not getting any phosphorylation of the STAT3. We don't see that with our Apex compounds. We then looked at in these models some biomarkers, and I'm kind of skipping ahead because we got these biomarkers from doing our single cell RNA-seq, but we looked at biomarkers that had been known to go down when you blocked REF1. So we looked at 2009 and NAPA and 9 plus NAPA, and you can see that they do go down and they're actually more effective even in the combo. And these are biomarkers published in the literature that are specific for NAPA alone treatment. And actually they went down, but weren't that great. They really weren't that dramatic. So we're not really sure if these are really true good NAPA biomarkers. And again, we can talk more about that. We're looking into that. So bladder cancer, I have to show you one, and this is interesting studies with Travis Jurdy in pharmacology and Dr. Fischel and myself, and we've been looking at the different stages of bladder cancer. Bladder cancer is interesting. They get cisplatin treatment. They do have some immunotherapy going on for it, but there's a lot of the patients become resistant to all the treatments. And so we're looking at new molecules to treat the bladder cancer. And this is a study, and I'm just showing you, these are actually from in vivo treatment of these T24 cells here, where there's a high level of APE1 expression, much higher than in the low grades and much higher than in benign tumors, high level of APE. And you can see that we treated with 3330 in 2009, just an example tumor. We got this very nice pattern of more potency here. What also is interesting is notice how red this is and not as red here. We're looking into the angiogenesis markers. Remember, we block HIF-1α, which would block VEGF, and so we think that we're actually blocking angiogenesis. So this is another tumor type that we're working on quite vigorously with our collaborators. Leukemia is an interesting story because this is a story with Ruben Kapoor. Ruben is actually now the director of the Wells Center here at IU in pediatrics. And he works on a lot of different signaling pathways in pre-leukemia and leukemic cells. And we got talking about, I gave actually a talk, and he said, you know, we really have this model that STAT3 and NF-kappa B are important, and in this particular model, SHIP2, are important in inflammation driving this kind of progression from a pre-leukemia to a leukemia state. And I'm putting this up here because I did nothing but supply the drugs and some expertise, I guess, and work. But again, you see this is familiar with the IL-6 receptor, signaling through JAK and STAT pathways. And so it falls in our bailiwick. So what Ruben did is he took our drug, 3330, and looked in his system to see if he could block really the progression of the cancer and really from a pre-leukemia to leukemia. And what you see, and this is with a TET2 mutated kind of an epigenetic model, you get pre-leukemic mice have certain levels of these leukemia, or sorry, blood markers. And upon stress, you get aging induced, and you get a lot of this elevation of the protein levels. But if you lose TET2, you get really a high level of this, causing a lot more of this progression from this pre-leukemia to more of a leukemic model. And when he did treatment with our drug in vivo, shown here, is he saw the return to normal in a number of different cases of the white blood cells and neutrophils and some others, the hematocrits and various things. And so this is quite exciting because you're actually kind of preventing this leukemia from developing and getting worse. Now, prevention studies are always hard to do in the population, but Ruben's looking at this now in some diabetes models, because we know that diabetes has high inflammation, including NF-kappa B and others. And so we think that maybe the drug may be useful in preventing the progression in certain subsets of populations of people. Whoops, I don't know why that's here. Oh, I forgot to block that out. So those are some of the things we're doing. As you'll see as I go through this, we love to collaborate. We collaborate with a lot of different labs across the world and across the campus. So, you know, with Melissa and pancreatic cancer and some other folks around here, bladder with Jurdy and some others, leukemia with Ruben Kapoor, chemotherapy-induced peripheral neuropathy. I'm going to show you a little data on that with Jill Fernbacher over in pharmacology. So we are really enjoy it. This actually makes the work a lot of fun. Oops. So before I do CIPN, is there any questions, or is there anything I could address? Because this is going to take a little turn from here.
Yeah, we do have one question in the chat, and I can read it to you, but you let's see. It's although tyrosine phosphorylation of STAT is the major regulatory modification, serine phosphorylation has also been shown to be important. Presumably, the slide you showed with no change in STAT3 phosphorylation was based on tyrosine phosphorylation. If that's true, have you considered additional assays of serine phosphorylation? And they said, great talk, by the way.
Oh, thanks. Well, thank you. Well, so to answer your question, you're absolutely right. That was the tyrosine. I don't think we've looked at the serine. We need to look at that somewhat, but you're absolutely right. That's a really good question. I will circle back because some of that was done by Melissa's group, and she may have looked at it and I didn't even know. So I'll look at that. And those, oh, same question. Okay. All right. So yes. Okay, we'll leave that for now. So now,
oh,
go
sorry.
ahead.
Well, we have another question in the Q&A.
Okay.
So, and it's with regard to the gene and evolution, are both functions found in this gene?
I got it. Thank you. Thank you so much. I love that question. So, so it's interesting. APE-REF1, the repair activity, APE1 is in the exonuclease 3 family that you find in E. coli. Okay, and it's conserved through organisms all the way up to humans. That's just talking about the repair. You don't get the redox function until you become a mammal. And we used to say just mammals, but I think there's now, if I remember right, there may have been, I'm blanking on another vertebrate that had it that wasn't a mammal. Because I used to say it wasn't enough just to be a vertebrate, you had to be a mammal. But it's a new function, it's a gain of function. And what's interesting is we actually published a paper on zebrafish, the zebrafish ape. Zebrafish ape has crystal structure and everything, looks very much like human ape. Almost you can lay them on top of each other, except for about the 30 to 40 amino acids, which are very disorganized in all the ape molecules, which we think are actually involved in some protein-protein interaction. But the interesting thing is the zebrafish ape has 6 cysteines. Human ape, mammalian ape has 7 cysteines. The one that zebrafish doesn't have is that cysteine 65, which is actually a threonine in zebrafish. And we took the zebrafish ape and we changed the threonine to a cysteine, and it gained the function of the redox activity. So it's actually almost a natural experiment showing that that cysteine 65 is the heart of the redox function. And actually, I'm glad you asked that, because I think in some of our redox assays going forward, we may use the zebrafish protein as a control too. So it's a gain of function. And so you could ask why, who knows, right? But I always like to wave my hands and say, well, mammalian cells, are under all this different stress, and you want to have tight signaling, and you've got reduction, oxidation, and everything going on. And so you really wanted to have ape hung out in the nucleus, doing repair, and it gained this cysteine that made it active to be able to convert through a electron transfer to turn on the transcription factors that are turn them on and make them or turn them off as need be and control them. And so maybe it evolved that way, but we really don't know. But it's a great question. All right, so and we have lots of papers on ape and cancer, its role, its redox role. So we got studying a number of years ago. I got interested in looking at APREF1 in the neural system, in either the central peripheral nervous system. And we actually submitted a grant, started thinking about, well, I think late onset diseases, neurological diseases like Alzheimer's or Parkinson's or ALS, some of these are probably an accumulation of DNA damage. And we thought oxidative DNA damage, because you're going through this whole life and picking up these point mutations. So we proposed a grant and submitted it to some neuro study section, saying we're going to look at APREF1 and its role in various neural diseases. And it got slaughtered, just absolutely killed. They hated us. They hated everything about us. But we went back in with the response, and it got killed again. And we talked to our program officer and said, gee, I really think, we're on to something here. And she said, you're trying to turn the Queen Mary with a robot. People are not ready to think this way. Go back to your cancer folks and see, how you could work on this. And so we did. And we started talking to the physicians, and we said, so what are issues you have with your cancer treatments? And all of them, without question, said, one of the problems we have when we give patients a number of drugs, taxanes, platinums, vinca alkaloids, is we get chemotherapy-induced peripheral neuropathy, and which is defined here on this slide. And we looked at this, and I was working with Michael Vasco at the time in pharmacology, and he's an expert in peripheral nervous system. And we said, oh, well, these are what neuropathy is. So these are induced by chemotherapy. And so we went back to the drawing board, did some experiments, and actually then we're able to get funded and actually have 2 grants now funded in this area, and one with Jill Fernbacher, both with Jill Fernbacher now. Just as on the red on the bottom there, and I was part of this recent panel on CIPN, there are no clinically effective treatments really for it, no FDA approved drugs, even though it's a major problem with patients coming off their treatments early due to the neuropathy. So what we think has happened, and I'm kind of giving the punchline, is we think that the redox function is not as critical when the cells, neuronal cells, are challenged with damaging agents as the DNA repair is. And we have some data showing that. And we actually got into this too by taking our drug, which I'm just trying to convince you blocks the redox signaling. And what we saw was an increase of DNA repair in the neurons with this drug. So how can that happen? And what we think happens is that by blocking the redox function, that protein-protein interaction, you're freeing up the protein to do more of its DNA repair activity in the neurons, but not in the tumor cell. And we've published that. So one of the studies we did, and actually we did this with Karen Pollack and the neuroblastomas in the animal core here in her lab, and we put in human neuroblastomas, mouse with human in the mice. And then we treated with cisplatin, shown here. This was modeled off of what patients would be getting their treatments every few days for 3 weeks. And we gave our drug, it goes by E3330 or Apex 3330, I can tell you why in a minute. And we gave our drug at the same time, and then we looked at various endpoints. And one of the endpoint we looked at is a marker for DNA damage, the phospho-H2X. And what you can see out here is that you see no DNA damage in the presence of our drug, and that's quantifiable. We also looked at this withdrawal behavioral responses, and this is just some of the data we have. So what happens is you can see if the mice are sensitive to putting these little metal rods kind of, and if their paw, if they pull it away faster or slower, and the neural folks have a way to measure all this. And what we saw is in the presence of our drug, it returned to baseline levels that they weren't as sensitive. So you reduce that sensitivity. So we had this nice in vivo data for this. So keep that in mind as I tell you something later when we talk about inflammatory bowel disease. So what have I said so far? We know blocking the redox signaling pathways are important in cancer. We know the drug blocks that redox signaling activity. What's interesting is in neuronal cells, what happens is now you're still blocking that redox function, but we're seeing a positive effect of that in the neurons by increasing the repair and reversal of oxidative DNA damage in the neurons. So it's really a nice drug, right? You're affecting the tumors, but you're protecting the neurons. So just keep that in mind, and I'll get back to that a little later. The next part I want to talk about is actually some bioinformatics, which is why you're all here, and we'll try to go through this a little bit. Any questions? Are we okay to keep going? Okay. One of the things that got us started on this path, and Nadia remembers this very well, is that when we were knocking down APE using siRNA, if you knock APE completely out, mice are lethal, day 6 or 7, cells are lethal, because if you think about it, 2 major functions, right? Redox signaling and DNA repair, oxidative DNA repair, which is very important. So by doing single cell RNA-seq, we were hoping to look at the heterogeneity and look at the different populations where here you have APE completely or way down, where you can't really see it, and other cells where you do have APE present. So this was our original goal, to do single cell sequencing. And I have to say, we kind of failed at that a little, because our knockdown actually, when we actually did the experiment, was a little too good. And so most of the cells had a lot of APE knocked down. And so we have to circle back to this at some point. But we did it both under normoxia and hypoxia, and we got a really nice separation. This is with Chi, who did this work, with Melissa, actually a lot of this in pancreatic. This is in pancreatic tumor cells. And we looked at the different levels, the population under hypoxia, the scrambled and the SI, and normoxia scrambled and SI, and did a whole bunch, and they've actually published some papers on this, but we saw a really nice separation of these populations. Well, the interesting thing is, well, the usual suspects came up that were affected. There were a lot of pathways that were the STAT3, HIF, and NF-kappa B, which is actually particularly a HIF under hypoxia, which was reassuring because we knew those were affected. So these were almost became are positive controls. But we didn't really want to look at what we knew. We were trying to find what we didn't know. So one of the things we did is we just picked a panel, I think about 12 genes, and went back to validate the RNA-seq to see if what we saw in the global effect. And we also went back and did this, not doing single cell RNA-seq, using populations of cells to see if we'd see the same change we saw in the single cell RNA-seq. And this is actually was published a number of years ago now. 10 of the genes were supposed to go down and 2 were supposed to go up. And we got a really nice correlation, a really nice data showing which the genes that went down and went up when we knocked down APE, over 80%, 80 to 90% knockdown shown here. So this now is what we kind of call our 12 gene panel. We now use this in different cancer cells because it's a validated a set of genes we can look at just to see if different cancer cells act the same, and actually normal cells. We also, but remember, when you knock APE down, you're blocking the redox and the repair function. So did those genes change because of the repair or redox? One way to get at that is to use our drugs that are specific for one or the other. In this case, we did 3330, and we got a really nice dose response even with this, with 4 of the genes we looked at. We just picked 4 and ran these. And so our argument is that these genes in this panc tumor are going down because they're under APE1 redox control. So that may mean there's transcription factors upstream of these genes, which we looked at, which are in our pathways that are under the control by APE-REF1. But these are genes that no one had ever really looked at before, being under APE1-REF1 control. We then looked at the pathway enrichment and which genes were down and which were up. And what really started to stick out is the TCA cycle glycolysis, OXPHOS, and then some metabolism pathways. And this had really never been seen. Here's even HIF-1α regulated genes that went down. We didn't really focus on the upregulation. It's interesting, a lot of the genes involved in replication in the cell cycle actually went up. But we wanted to look at this because this was new. This had not been discussed before when you modify APE levels in cells. And when you looked at the genes that were changed, we started looking at, in the different pathways, and we focused on the mitochondria due to the glycolysis, TCA cycle, and OXPHOS. And I know this is probably hard to see, and the colors indicate the level, the density of the color indicates the level of down green and up red or pink. Up, pink is up. And you can see really all the complexes are hit in the mitochondria. Remember, these are genes mainly nuclear encoded that go to the mitochondria. So we're regulating these genes, and this under normal oxygen. And what is always striking to me is if you look at this and this is under hypoxia. And you can see that it's more intense, a lot more gene changes, and it's more dramatic. And I always like to just shift back and forth, and you can just stare at the this and the impact. So it appears that blocking APE-REF1's activity under hypoxia even had a more dramatic effect. Not necessarily surprising, but was interesting nonetheless. So, and again, I apologize, this is actually in a forthcoming paper. We started looking more intensely at hypoxia and normoxia, and we looked at the glycolysis, pentose phosphate, the different metabolism pathways. And again, green is down and red is up. And actually what you see is there's more intense changes here in the hypoxia versus the normoxia. But you can see that we're impacting a lot of the different players in these pathways, not just 1 or 2. There's a lot of different ones. So we've been spending, this is work particularly done by Shilpa, postdoc, in Melissa's lab, and we're contributing some to this. But we got very interested then in metabolism and what APE1 is doing. So we, again, picked some genes that were shown to be changed from that pathways I showed you before in different complexes. And what you see is that it goes down in both normoxia and hypoxia when you do the knockdown single cell RNA-seq, and then when you validate it, looking this in the population of genes. So this was reassuring that some of the genes, identified here with a deeper analysis, again, showed very much when we validated that. We also started to look at the different pathway now, and it's been decades since I took intro biochemistry and the TCA cycle and everything, and unfortunately we're having to relearn all that. But we started looking at some of the different major players in the various metabolism pathway, and this is using one of our new compounds, 2009. And again, we get a very nice dose response of our drug knocking down these main players in the cycle I'll show you here. So really, we're affecting this part of the TCA cycle with our drugs, okay? And the point I want to get to, and we have a lot of work to do, and I'm showing you just some of this parts, but since we got into this, we do what we usually do. We started scanning the literature for things we could do in combination with our drugs in metabolism. And there's a drug out there called CPI 613. Its new name, they call it devominostat. And it inhibits PDH and KGDH, which is involved more on this part of the TCA cycle. And since this is in clinical trials, we like to use drugs that are actually been, validated in clinical trials for what that's worth. And we started doing some studies to say, I'll bet if we combined this drug with our drug that's affecting this part of the cycle, we would see some combination effects. Obviously, we got the result, or I wouldn't be telling you about it. And so what you see here, and these are plates. This is a neat system called from Biolog. These are mitoplates. These are actually S1 mitoplates, and they're substrates of the different parts of the TCA cycle that have a dye with them. So you can just look. You can run these. So you can run. Here's scrambled or SI APE, and then each one of these rows, there's controls, and each one of these rows has different substrates. And then you can look at the change, in the intensity and measure that. So it's a functional assay of the mitochondrial complex and electron transport. And it's a really cool assay. And we've been doing a lot of this. And what you can see is with the knockdown, you get an effect of these four main components of the TCA cycle. With 2009, you get a really nice effect. Devominostat, a little bit, not as much. Up here, this molecule is actually made with a grad student at Purdue, and his name is Rodney Nairn, and that's why it's RN758. Unfortunately, this was one of the molecules that was dead, that had no activity, but had the same similar chemical structure. So actually, it lives on as a negative control, which is really, really important. And frankly, negative controls can be as important, and you can see no effect. So we use that all the time. And these were in calves. We didn't really notice really not the same effect in the calves as we see in the tumor. And that's the other interesting thing, is we're getting a tumor effect and not a calf effect. So we can look at that. We then looked at combos using 2009 or the devominostat, and again, looked at genes, mitochondrial genes, and we saw this combo effect. We looked for genes regulated by REF1, and we actually did see a combo effect there. And this is just showing then the combo when you look at the functional assay, you can see here. So we've done a number of these things. And this preprint's actually online. If you're interested, I can send you the link to that preprint. We hope to get it back in very soon. But this is the point I'm trying to make, is we took, everyone is stuck on APREF1 and what it was doing, and all of a sudden it's just exploded now that we're studying a lot in the mitochondria, the metabolism, and it was all from our analysis of the bioinformatics that came out, frankly, with the studies that started with Nadia and then have gone from there. Okay, so let me move on now very quickly to go through translating this into the clinic. And am I doing okay on time? Are we okay? Okay, so how do you take this to the clinic? So it's a long road. Like I said, I've been working on this. I moved here in '93. We were working on this way back in the '80s. We actually made the first antibody to APE1 and licensed it to Novus Biologicals. It was their first antibody they ever licensed and sold, and they've, I think, since been sold. But the antibody they sell is still our antibody. It's monoclonal that we've made. And then we've been working over the years. The red part is my academic life, where we've been looking at different things and the transcription factor activation, different cancers. We did some work starting in 2008 and then 2011 in the eye, which I'll talk about. And at the time, in about 2008, we formed a company. It was called Apex. At the time, fabulous time to start a company, because in 2008, 2009, the market crashed. And so everyone went running for the hills. So it got very hard to get funding. But luckily enough, there was a group here in town, Pearl Street Ventures and some others, who put some private equity into the company. And we got our an SBIR in 2013. But we've been working on this academically and then even from a company standpoint for quite a while. In fact, it even continues. It takes 2 slides to show, is that we renamed the company, had additional funding, and we took an approach that we said, look, we know a lot about 3330, and we are making new compounds. What's the fastest way to get to a trial? Now, 3330 had a life before I got a hold of it. In the early 90s, it was developed by Eisai in Japan. Eisai was looking for inhibitors of NF-kappa B, and they were studying hepatitis and inflammation. And they did, and you know, Eisai is a big pharma, and they screened lots and lots of molecules. And the one that they found that they took to phase 1, phase 2 trials, and they did 10 phase 1, phase 2 trials in Japan, never published, is 3330. It was called E3330. They thought it was binding NF-kappa B. As it turns out, it isn't. It's blocking APE's ability to turn on NF-kappa B. So they were kind of in the right church, wrong pew, if you will, and with that. So they took in the hepatitis trials, they saw it was very safe. They did phase 1, phase 2, they did PK, and they did all the workup. And about the time they finished their phase 2 in chronic hepatitis C, and they saw efficacy, some, they in-licensed Humira and an antiviral drug. And they just put it on the shelf, the patent expired, and it was done. Well, since we were working on APE and then REF1, you know, I told you the story with Tom Curran in the 90s, discovering the REF1 activity through some various literature searches and patent searches, I saw this molecule. And so we got some 3330 and synthesized it, also actually up in Michigan, and started using it as a molecule to study the REF1 activity. And that's what I've been telling you about through the first part of this talk, and then made new composition of matter agents, 9, 14, and some others. So one of the things my company knew is that it's been in humans, it's been safe, we know some starting doses, and there were no adverse events. So we took to the FDA, we actually got from Eisai, we had a partnering deal, and we got all of their data, preclinical and clinical, to us. We had to translate it because it was in Japanese at 25 cents a word. And but we were able to put together a package to the FDA. And because we then synthesized our molecule exactly like Eisai did, we had it made the same way, we used the same columns, we did everything the same. The peaks from the HPLC and mass spec and everything were exactly the same. It's essentially the same drug. So the FDA accepted all of that data that we had translated to use in our application, and it got approved immediately because it had been in over 300 patients. So it was very safe. So we started our phase 1 trial, and we started it in solid tumors. And we started in 2018, and we thought it would be about a 2 or 3 month trial, but it turns out some patients stayed on over a year. And so this is the trial that we presented at ASCO in 2019 on using this in solid tumors. And so this was 19 patients, phase 1, it's a safety trial. We dose escalated from 240 to 720 milligrams. We got to 720 milligrams. We saw a slight rash. And so we reduced the dose to 600. It's an oral drug, and the rash went away. We had patients that were on, one patient was on 421 days, one close to a year, and another one also close to a year, and one 252 days. We saw we didn't see any tumors going away. Remember, these patients had to be on the drug, only this drug, no other drugs. They had failed all other treatments, and they had to have tumor that was progressing at the time of going on the trial. So the tumors were not static, they were growing. So we were able to stop in this patient here, for over a year, the tumor just stayed static and didn't grow. Again, because of the mechanism of action, you wouldn't necessarily expect the tumor to disappear. But what we were also able to do is do the PK on these patients. We were able to then, for 3 of them, we were able to take biopsies on the tumor before they took our drug and then after we took our drug. And so keep your eye on the green arrow, the melanoma, and then the 2 red and pink. So these 2 patients did not stay on the drug very long before their tumor progressed. And this patient stayed on quite a long time before the tumor progressed. And what we did is we did some proteomics, actually here in the proteomics core, and some other things, but I'll show you the proteomics data. And what you can see is that the shift in the melanoma, the protein levels changing, was very different between the panc and colon from the melanoma. And if you look at the depths in this, you can see that panc and colon were very different pattern than in the melanoma. And, and we're doing more analysis on what these genes are. But what was interesting is if you look at genes that are downstream of HIF-1α, NF-kappa B, or STAT3, we saw that they changed. They did change all the patients that were on the drug. So that's sort of a PD, a pharmacodynamic, showing we did have target engagement, and the genes were regulating, genes downstream of it went down. What was surprising is APE, in that one melanoma patient, the person who stayed on for a long time, it actually went down. And the drug, remember, doesn't, we're not blocking APE's expression, we're blocking its activity. So what we think happened was when the tumor wasn't growing, you didn't need as many transcription factors turned on. And so you don't need APE to be revved up as much. So again, an N of 1, this is a phase 1 trial. This is just some initial data. We also saw APE protein levels in the stable disease patients who were on longer. The protein level of APE in the serum went down. I have no idea what that means. Less APE being made. We do know APE gets shed. There's exosomes that have APE. So could it be a potential marker down the road? Again, too small of a study to make a conclusion. And the PK is very reminiscent of what ACI saw. This is all the different dose levels, and you get it, it goes up and it stays pretty stable over time. And this is just looking at the 600 mgs a day. They got it 300 mgs twice a day. This is just in the short time points of 1, 2, 4, 6, 8 hours. And then you can see it just stays very stable over time. So you can get stable dosing of the drug. Studying it in cancer, we did a phase 1 trial. We see we can hit some of our markers. It's very safe. And remember, we're hitting HIF-1α and NF-kappa B. And what is that important in? That's important in some ocular diseases. And so one of the areas I want to talk about very quickly, am I still okay on time? Or, okay, I want to leave time here, sorry. I'll try to get through this quickly. So these are the 4 pieces of the pie we're kind of focusing on. I've talked to you about the CIPN. I've talked to you about cancer. I want to talk about these 2 pieces now, particularly as they relate to trials. So new directions. So diabetic retinopathy is, and diabetic macular edema, very important, large patient populations. Diabetic retinopathy is an early stage of the disease where you start to get the blood vessels with leakage and growth. You start to get your vision blocked. There's really no good treatment for it right now. If you have age-related macular degeneration, the wet version, that's a lot of angiogenesis going on, and patients get injection of their eyes of Avastin, bevacizumab, which is Avastin, and Eylea, which is and so that you have to come in for monthly injections into your eye, and it's an antibody to the VEGF. It works pretty well. Patients become resistant, and people who have diabetic retinopathy are not going to come in because their vision isn't going yet. They're just starting to show it, and you're not going to come in for monthly injections until it develops. So this is a really good area to look at, is how we could prevent this from going forward. Diabetic macular edema is another kind of early stage. And this is what it looks like if you have DME. And this is what we think it'll look like if you can prevent that from occurring, is that your vision would be much cleared up. So why 3330? When we published or had our abstract at ASCO, we got a number of eye companies approached us and said, you know, you're really in our wheelhouse. We want to block inflammation, which we know is involved in these diseases, and you want to block angiogenesis, which you've clearly shown that. So through a course of a number of discussions over a year, one company, Ocular Pharma, actually this is their slide here, they licensed the drug from my company to Ocular, which is now a public company, to do trials in diabetic retinopathy. And they were excited because, again, we're blocking both the angiogenesis pathway and the inflammation pathway. It's a novel drug, novel target, but you're still blocking pathways that ophthalmologists are familiar with, so they're comfortable with. It's a new molecule, it's oral, so you can take this. And remember, I showed you the PK where you can get the stable amounts in the serum. That's important because you can keep a nice stable level of the drug. And it's been shown to be safe now through lots of patients, both in hepatitis and oncology. The data we have, and this is data with Tim Corson, we'd published other data with an injectable form. This is giving oral dosing of 3330. And what you see is a reduction in the amount of lesions that occur. And this is a staining for the lesions shown here and then here when we give our drug orally. And the level you can get in human is much higher than the level that you need to have in mice. So we think it's set up really, hopefully, for success. And this is the trial that started in April. It's 100 patients. It's 50 placebo and 50 on the drug. It's going to be 15 to 25 sites. And it's a phase 2. And it's looking for if you get this a two-step improvement on the scale that the ophthalmologists use, and then we have some secondary markers. So we think the readout for this trial will be in early 2022 because it's a 6-month trial on the drug. So we're very excited. And again, I'm just trying to tell you how we're following the data, and following the science, and following the disease indications, which takes us to the other one we've been excited about lately, and it's inflammatory bowel disease, which is Crohn's and ulcerative colitis. It's a huge number of people have this, a large market. Most of the drugs, you take Humira and other things, you can take steroids, but they usually have a lot of side effects, and a lot of colitis, not a lot. Some of the ulcerative colitis will eventually progress to colon cancer. So this is an area where there's a lot of research, and they think it's due to inflammation, and a lot of work now, and a lot of large pharma starting to look at neurons effect. There's myenteric neurons that are in the bowel that are involved, and they think there's an interaction between the epithelium and the neurons. Okay, now remember, myenteric neurons, those are peripheral neurons. So what does that ring a bell? Dorsal root ganglia are peripheral neuropathy studies we did. So I'm trying to bring this back around. So in a healthy colon here, if you start to have dysfunction, oxidative stress, inflammation, it leads to chemokines, and you end up with your inflamed colon. And we think it's through the interaction with the myenteric neurons with the various inflammatory cells in the epithelium. So, and this is in a publication that just came out. The model we use, and this is a collaborator in Australia, she has this Winnie mouse model. And what happens is if you look at the genes, expression of genes in the Winnie mice compared to IBD patients, is that they're 91% concordance in the UC models, and about 88 and 63 in male and female with the Crohn's. So this is a really nice genetic model where you're getting the same genes in the mice that are showing up in patients. Well, I love this slide because you don't really have to know much to say, here's the control mice, here's the Winnie mouse, and here's with 3330. And I think you can see that there's less rectal prolapse in the blood vessels, and you'd much rather be this mouse than this mouse. And so I think this is just a picture's worth 1,000 words, but that won't stop me from saying 1,000 words. And so if you look here, if you look at the normal colon, you look in the Winnie sham mice, how they look disrupted, and you look out with 3330, they look more back to normal, and you can quantitate this. You can also, and the cool thing about the model system is they take the colons out and do contractile activity, and you can see and get these patterns of, this is the contractions, and here's what the Winnie sham looks like. It's a mess, and look what happens when you give our drug. It looks almost the same as control. And because we're interested in the neurons, if you looked in the myenteric neurons, and we looked at mitosox, which measures the oxidative stress in the mitochondria, control, Winnie sham, and it's almost all gone by blocking this. And my favorite is actually, if you look at the neurons and you stain for the neurons, here's control, here's your Winnie mouse, and here's 3330. And she actually saw neurites growing back and growing out. So this is very exciting. And if you measure for oxidative DNA damage, control mice, here's your Winnie mice, and then here's with our drug. So you're blocking, remember, we're blocking oxidative damage here. This is adeoxyguanine. This is a marker for DNA damage, and it looks like it's being repaired here. So this brings us back to our neuron studies that we looked before in the peripheral neuropathy. And so we think that we can tie these different studies together. So where does that take us and leave us? That leaves us with, we have a number of different areas to look at more now in the metabolism, which came out of all the data analysis to look at that, the role in the mitochondria. We're looking at various combinations in cancers. And then it leads us to look at other indications like ophthalmology and IBD. And so we hope to continue these studies with more clinical trials in all these areas. Obviously, this one's going on. We're looking to do trials in these other areas. And with that, I'll stop. We have actually a small group. I think we're very efficient with a number of people who've worked in the lab on the last couple of years on these studies. And, we do a big shout out to Dr. Fischel's lab, and a couple of people in her lab have been instrumental in all of this. And obviously our labs are next to each other, so we've had a long-term collaboration. And then we like to collaborate with people. If any of this triggered anything and you'd like to collaborate, we're happy to do that. We have lots of the drugs, if you have good animal models. We've recently set up probably a couple more IBD collaborations that we're looking at that's come from this in other diseases. And then, of course, Nadia, I don't know why Nadia's name isn't on here, but Nadia started here early with the studies, and then Junwan, who Nadia works with in the bioinformatics core between IU and Purdue, and Dr. Chi Zhang, who's done a lot of the analysis with Melissa through this. So I think I've been, to conclude on this, I've been very fortunate to have really, really good collaborators, fabulous collaborators who we don't have problems with, who we're all working in different areas, been supported by a lot of people. We've had a fair amount of funding. We've been lucky. And it's a lot of fun because we're still interested in cancer. We're still doing neuropathy, but now we're expanding it to the IBD. There's still some more work in the liquid tumors with leukemia. And then of course the eye is very exciting because we have the phase 2 trial. So another trial we'd like to do, a couple of trials would be combine our drug with the injections for, say, wet AMD, because to see if you could extend the length of time between the injections by giving the oral dose of 3330 plus or minus an anti-VEGF antibody. With that, let me stop and take any more questions.
Thanks, great talk. You touched on this a bit, but I was wondering, so how long does it, I mean, I'm sure it's variable, but how long could someone expect to take to go from bench to translation, and even from like an indication to then a clinical trial? And then can you also mention a little bit more about that process? It's sort of like a black box to me.
Yeah, so I think it kind of depends where you start. I mean, if you're starting at discovery where you have a target or a drug and now you have to work through, it's going to take 5, 6 years, 7 years, and it's very money dependent, because you can do your studies and you have a drug and a target, and you do your in vivo work, and you see it looks good. Then you have all this called IND enabling. So then you've got to look at, and that's multi-millions. That's $2 or $3 million maybe, because you've got to make enough of the drug. You've got to make it under GMP conditions that you could give it eventually clinically. All the large animal, and in the IND enabling, you have to use, large animal studies. You have to do 24-day tox studies. So you have to have enough of the drug that's made in the same way you're going to give it to patients. So there's a cost involved in that. And then once you get approval, the IND is approved by the FDA. IND is a new investigational drug. You got to get the drug made. Are you going to give it oral? Is it IV? It's got to be made in that. And then you have to have sites to give, phase 1 isn't many patients, but it could be, we did it really cheaply. We were able to do it, but it's like $50,000 to $70,000 a patient. And so, 20 patients, you could do the math, and and that's just for a small trial, so again, it just depends on where you start, but you're looking at 5 to 7 years. Now, if you have a clinical drug and you said, okay, I want to repurpose it, then you can start farther along because it's been through all the FDA approval and IND enabling and all that. Now, you may have to do a small dose study because it may be for the indication you're going to look at it. Like if we give it, so actually in the eye, we're giving it at 600 milligrams a day, 300 milligrams twice a day. And we're starting at that because we know it was safe in the oncology with no side effects. But do we need that much? So after we finish phase 2, the phase 3 trial, assuming it's good, the phase 3 trial would probably be a dose response trial where we might cut the dose in half or cut it down to see what's the lower amount you could give daily to get the positive effect. Because if you're going to take this chronically, you might want to take the minimal amount. So you could do a dose range finding study, that sort of thing. So it just kind of depends on where you're starting. And it really, frankly, depends on the amount of money. You can go through faster sometimes with more money. So. Any other questions? Everyone's still awake? Is that cool? Well, if anyone has any questions or anything, I'm happy. I answer all my emails. So, whoops. Oh, thanks. Thank you very much. Oh, here's one. Limiting step. Hang on. Limiting step of drug development. Wow. It could be, that's a good one. Well, I think lots of, we all think we have the best targets. We maybe are developing a drug for our target, and we all think we found the best drug. I think the limiting step a lot of times is really in the PK, PD, in the pharmacokinetics, pharmacodynamics, showing your drug, works in vivo, and you have a good animal model. I think the other rate limiting is, do you have good biomarkers? And so I see in the question, they said, most drugs fail, and that's right. And everyone keeps trying to figure out, how do we come up with models? Obviously everything that goes into humans pretty much has worked in their animal model, right? So clearly they're not the best predictors. The other would be biomarkers. There's a lot of push now for AI to be able to crunch all the data and come up with targets, that are important in the different cancers. And it may be that you would design your drug, it may be a very small subset of patients, right? Where the drug would work on, and not, every panc patient, but maybe certain ones with certain biomarkers. So that may be also successful. We may see that you might need, and we know we're fractionating. Precision medicine is all about getting the right drug to the right patient. So you may end up having to develop different drugs for 30 different subtypes, right? And then that becomes, how do you get those drugs developed? Is there a market if it's going to be big pharma? A lot of those are going to be developed through academia, because we're interested in helping the patients. It's not driven by our stock. So I think there's just a lot of different places where the drug fails. I think a lot of it is in the modeling. A lot of people have drugs that they look good in all their preclinical, but they just don't have good, bioavailability when you get to the patients, or it's the wrong group of patients that got treated with the drug. So I think with more of the big data and bioinformatics and AI that comes in, again, I think we're going to be able to see, I think it's going to have, for all the ethicists and stuff, it's going to be very interesting. What if you find that there's just a small sliver of a cancer that could be treated by, say, your drug? What is the cost going to be? Who's going to develop that? We're going to run into that, I think. And I think that's something down the road that'll be interesting. What else? Let me see. Yes, that's fine. Just shoot me an email about doing any combinations. I really think the combinations are the way to go, clearly with our drugs. And our drugs, and I'll make a little pitch, our drugs are so safe that they're good to do combos with. So anything else? If you have more questions later, please feel free to email or. Anything? Okay. Thanks again, Min and Nadia and everyone, for letting me babble on for another hour and 15 minutes. So it's fun.
Yeah, thank you so much. And I'm sure we will get more exciting stories, as you mentioned, next year.
We hope. We hope we have. If we have positive, we'll definitely be excited about it. Yes.
Yeah, hopefully the COVID didn't really affect your journey that much, right?
We had about a month, and it affected some of our animal studies. We had to postpone to do those because of the LARC and the people not being able to come in as much. Frankly, we ended up writing 2 or 3 papers and a couple grants, and we actually published one of the papers I showed some data from was data we'd had for 3 or 4 years, and so it forced us to sit down and write the paper. So we were very fortunate. And we're fortunate everyone here's been healthy, so that worked out, for us doing that.
Yeah, great. Yeah, thank you so much. As you show the single cell picture, because we did talk a lot about the single cell data analysis. So it would be nice to see how this really, work in practice and help with the translational research.
Yeah, no, single cell launched all of this new direction. So we're excited. Okay.
Yeah.
Thanks again, everyone. Enjoy.
Thank you so much. And thanks everybody for coming today.
Thank you. Thanks, everyone. Bye.
Bye.